Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing paper 1 MCQ of A-Level Biology, October-November 2023, variant 13. Question 1 deals with light microscopy. It's stating what statement about light microscopy is correct. As the resolution increases, the magnification increases. No, there's no such relation there. As the magnification increases, the image always becomes clear. No, the image blurs up. The resolution will decrease. Yes. As the increasing wavelength is used. Yes, because they have an inverse relationship. Option C is correct. Magnification resolution are the same terms referring to same factor. No, that's incorrect. Question 2. The diagram shows the stage micrometer scale viewed through the eyepiece graticule containing eyepiece containing a graticule. So this thing is the eyepiece graticule. This is the eyepiece graticule here, and this scale is the stage micrometer. So we have the stage micrometer, the eyepiece graticule. The small divisions of the stage micrometer are 0.1 millimeter, which means this bold line on the stage micrometer and the other bold line on the stage micrometer, these are 0.1 millimeter apart. So this is 0.1 millimeter apart from one another. So once the image is superimposed, we see that 40 divisions of the eyepiece graticule equals to 0.1 millimeter. This is what I mentioned here. We just count the divisions here, 10, then 20, then 30, then 40. A total of 40 divisions on eyepiece graticule equals to 0.1 millimeter. The stage micrometer scale is replaced by a slide of plant cell. This is a plant cell shown. They have given to you a nucleus. They are asking you what is the maximum actual length of the nucleus in the plant cell. So what we'll do, we'll just measure the width of the nucleus, starting off from what I've done. Just draw the straight line and just count the number of divisions that it corresponds to. So it corresponds to 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, a total of 10 divisions. So what I've done, I just performed the simple calculation using the scale that I actually derived. 40 divisions equal to 0.1 millimeter. So 10 divisions is equals to x. The simple ratio method, you just cross multiply. X is equals to 0.1 into 10 divided by 40. The answer comes out to be 0.025. Now, since this is not in micrometer, it's in millimeter. What I'll do, I'll multiply it by 1000 to get the answer in micrometer. It's 25 micrometer. So this is my answer B. What is the correct order of involvement of cell structure in synthesis and secretion of enzyme? The organelles and cell structures are shown to you. We know from these, the nucleus should come first, then the RFER, then the Golgi, and then the cell surface membrane. And using this order, only option C is correct. Central cilia microvilli structure found in eukaryotic cell. Which statement about these cellular structure are correct? So whatever option you pick, all three should qualify for that. Which statement was cell surface correct? Central cilia and microvilli are composed of microtubules. No, it's not correct for cilia and microvilli. Centrioles play a role in mitosis and semi-conservative DNA replication. No, there is no role there. Cilia and microvilli both increase surface area. Microvilli increase surface area. Cilia have no role in increasing the surface area. Microtubules are made up of proteins that form spindle. Yes, that's correct. Fact. Which description of plasmodesma is correct? There are channels through the plant cell walls lined by cell surface membrane. Yes, they are lined by cell surface membrane. Channels lined by protein. No. Channels required for movement of water along apoplastic pathway. No, that moves along cell walls. They become lignified. No, they don't become lignified because they don't have cell walls. Which cell organelle does not have nucleic acids? So all these have nucleic acids. Golgi is the only one that lacks nucleic acids. Which process occur in eukaryote and prokaryotes? So in prokaryotes, we know they can never be mitosis occurring because they don't have a nucleus. So they divide by band division. So hydrolysis, transcription will occur. Mitosis cannot occur. So mitosis should be omitted out. So if you omit out two, so that means the answer cannot be A, B, or D. The answer will be C. Which bonds are present in all viruses? So all viruses should have covalent bonds. For ester bonds, no. All viruses do not have lipids. Those which will have lipids, they will have the ester bonds. Otherwise, others will not. Phosphodiester, yes, is present in the nucleic acids. So yes, again, the answer here is 1 and 3 only, which is C. Again, you can see that 2 was incorrect. So in this case as well, you can omit out 3 options right away. But for that, you have to be very sure which options 1, 2, 3 are correct and which are not correct. The diagram shows structure of a monomer. If you just go through this structure, where is it present? So you can see if you start to just see which monomer is there. So it is present in amylopectin, sucrose, and glycogen. Yes, it's present in all three. It's simply a glucose molecule, alpha glucose. So Coming on to a triglyceride, consists of cholesterol, consists of fatty acids, L, O, and P. These are the three names provided to you. The diagram shows one possible arrangement of the three fatty acid LOP in a molecule, cholesterol molecule with three fatty acid chains attached. How many different arrangement of the fatty acids are possible? So one arrangement is the LOP. The other could be like L comes here, O goes up, and P stays there. So this is the second arrangement. The third arrangement could be L goes up, O comes down, and P comes here. Other than these three arrangements, there is no possible fourth arrangement because we know that the molecule can rotate. Eventually, it will be the same. So it's just three. Which statement about phospholipid molecule is correct? They contain one saturated fatty acid. No, there is no such fact of one and two saturated and unsaturated fat. They contain phosphodiester bond. No, nucleic acids have this bond. They contain hydrophobic region and hydrophilic region. Yes, phospholipids are having both ends, polar and non-polar. So C is correct. They contain a hydrophobic phosphate group. Phosphate group is correct, but it is not hydrophobic. It's a polar end. Hydrogen bonding explains why many properties of molecule water, including high latent of vaporization and high latent capacity, high specific heat capacity. For which processes in plants is hydrogen bonding is in water important on hot sunny days. So the specific point here is a reference to hot sunny day. It would prevent denaturation of enzyme in leaves. Yes, on a hot sunny day that's possible. Reducing water loss by evaporation. Yes, on a hot sunny day this can happen as well. Allowing leaves to cool down quickly at night. Now there's no relation here, so we can skip 3. Holding the column of water and xylem vessels together. Yes, hydrogen bond is relevant there as well. So 1, 2, and 4 is the answer. Again, since 3 was omitted out, so that means that answer A, C, and D are not there. B will be the answer. Which feature of cellulose molecule contribute to the function of the plant cell wall? Adjacent cellulose molecules are linked by a glycosidic bond. Adjacent cellulose molecules, no. Adjacent beta glucose molecules are actually linked. Branch chains of beta glucose molecules are linked by hydrogen bonds. No, cellulose is not branched. Plus, they are saying branched beta glucose. Wow. Anyhow, students have these habit of mixing words, so they play with these tricks. Molecules of alpha glucose are linked by avances glycosidic bonds. No, one alpha glucose is not there. Unbranched chain of beta glucose are linked by hydrogen bonds. Yes, that's possible. That's actually how it happens. Which statement describes some of the enzyme action? Enzymes hold reacting molecules so that their reactive groups are close together. Yes, that's how the reaction proceeds. That's how the catalysis will proceed. In an enzyme catalyzed reaction, more molecules have sufficient energy to react than without enzyme. Yes, that's how the activation energy is lowered. That's why more reactants proceed to produce products. Reactions catalyzed by enzyme take place at a lower temperature than they would without enzyme. Yes, that's possible because of the activation energy has been lowered. So all three options are correct, 1, 2, and 3. Influenza virus has an enzyme called neuraminidase, which breaks down glycoprotein in the surface membrane of the cell that virus infect. The glycoprotein bind the active site of neuraminidase by induced FIT. Which is about induced hypothesis of enzyme action are correct. The active site must be of the same shape. First of all, it's never of same shape, it has to be complementary. So same shape is not possible because this is induced FIT. So it should be complementary, but again, slightly different. The enzyme is less likely to be affected by non-competitive inhibitors than an enzyme working by lock and key. No, there is no such info provided to us. The substrate is converted to product by a specific R group in the active site, just like the lock and key mechanism. Yes, that's very well possible because it's always a specific R groups in the active site that actually are involved in catalysis. So only option 3 is correct. The graph compares the effect of temperature on the activity of protease enzyme papain when solution free and when immobilized in alginate beads or alginate beads. Temperature is provided on the x-axis. Activity of papain is given there. You can see that the activity is high initially at lower temperature, but as the temperature increases, the activity goes down of the free papain. Whereas for the immobilized papain, it takes a bit longer to become denatured and the activity gradually decreases. Which is statement about the effect of immobilization of papain is correct? It alters the shape of papain's active site? No, if active site was altered, then the reaction would not take place. It decreases the activity of papain? No, it increases. It's actually not decreasing it. There's no evidence there. It increases the stability of papain. Yes, that's why it actually had structural support from the alginate beads. That's why it stayed firm and its active site did not get distorted as easily. It reduced the number of collisions. Again, there's no evidence in the graph to support this. Plus, as we increase the temperature, the collision will definitely increase because that's just this basic physics as well as chemistry concept of how kinetic energy increases the collisions because the molecules will collide more often. Question 17. The diagram shows the adrenaline activates the processes inside the cell. This is the adrenaline molecule. It's a cell signaling molecule. It being attached to a protein receptor. Here they are calling it as a beta adrenergic receptor. It's on the cell surface membrane. And then there's a G protein which become activated, and inside the cell a cascade of events will take place, which eventually will lead to the enzyme being activated. Conformational change will occur, we know, on this receptor once the adrenaline attached. So the G protein can go and activate the second messengers. This is the fact that we already have studied. Which row is correct? Type of receptor molecule. The receptor molecule can never be a phospholipid, it has to be a protein. So either the answer is B or D. Results of ligand binding to the receptor. Receptor changes in shape. Yes, receptor leaves the cell membrane. No, the receptor cannot leave the cell membrane. It stays there and its conformational change occurs, it changes shape. So B is the correct answer. The statements are comparisons of endocytosis and exocytosis. Both are mechanisms that involve vesicles or vacuoles or transport of material across the cell surface membrane. Yes, that's the correct option. Both mechanisms occur to allow bulk transport. Yes. Endocytosis involves taking material into the cell as well as out of the cell by exocytosis. Yes, that's correct as well. Some of the cell surface membrane is lost in endocytosis. Yes, because the membrane pinches in, so some surface area will decrease. And it increases upon exocytosis because yes, some membrane fuses. So yes, all four statements are correct. 1, 2, 3, 4. You count the number of statements which are correct. A student was asked to calculate the surface areas to volume ratio for an agar cube with a side length of 5.5 millimeters. Side length is provided. We know that for cube, this is how you calculate the surface to volume ratio, 6a squared upon a cube. So the value of a provided to us is 5.5. So you compute it in this formula. The answer comes out to be 1.09 upon 1. Or simply, the formula becomes 6 upon 5.5 because both a square and a cube are there on one top of the other. So it's just 6 upon 5.5. So again, if you just simplify it, it's 1.09 divided by 1. So the ratio is simply closest to D. Which is statement about behavior of chromosome during telophase is correct. Chromosomes attached to spindle fibers at the equator. No, that happens in metaphase. Chromosomes start to uncoil in the newly formed nucleus. Yes, that happens. Chromosomes move toward opposite poles. No, that happens in anaphase. Chromosomes condense into compact structures of cytoplasm that occurs in prophase. The answer is B. Transmission electromicrograph of shows a cell in stage of mitotic cell cycle. So you can see the cell is like about to just go, like still become divided. So this is telophase occurring. We can see the cells forming a furrow here, and it's pinching inwards. The chromosomes are being pulled to the opposite ends. So that shows this is anaphase as well. So which is to explain why it's difficult to identify the stage of the mitosis cell cycle shown. Chromosomes are supercoiled. No, chromosomes are not supercoiled here. Anaphase may be continuing and telophase may be starting. Yes, both can be seen. It's unclear whether the electron micrograph show two cells. No, it is very clear it's two cells, but they are not at metaphase. Some people may consider interphase to have started. No, how can interphase start? Because without the cell undergoing cytokinesis. So if two cells actually were there, separated from one another by cytokinesis, only the interphase would have started. During lagging strand replication, short fragments of DNA are produced. The fragments are joined together by DNA ligase. We know that basically is caused by the enzyme DNA ligase. Okazaki fragments are joined together, which should correctly describe the structure of these short DNA fragments. Have bases sequences that are complementary to the newly strand of leading strand? Yes, that's correct. So A and B cannot be the answer. Either the answer is C or D. Consists of one polynucleotide strand? Of course, yes. So again, with this, we know that C is not the answer. The answer would be D. Contain only carbon, hydrogen, oxygen, and nitrogen? No. The DNA must contain phosphate group as well. So a cross should be there. So that's how the answer is D. 23. The genome of a bacteria, Escherichia coli, has been altered to enable it to code for an amino acid that is not found in nature. All the ATC DNA stop triplets on the DNA strand that is transcribed have been substituted to ATT. The ATC triplet can then be inserted to code for a new amino acid, so it is inserted back. So this is what we insert. A new tRNA can be constructed to carry out amino acid synthesis. So what is the anticodon of the new tRNA? So the coding strand is having ATC, the mRNA would be UAG, and for this mRNA codon, the anticodon should be AUC. So that's simply complementary base pairing. So this is mRNA, and this is tRNA, and this is our coding or the template strand. So if we just follow the simple rule of how complementary base pairing occurs, so that's AUC. 40% of the bases in this section of a non-transcribed DNA strands are purine molecules, which means adenine and guanine are 40%, so 60% must be pyrimidine. That actually is what they're asking. What will be the total percentage of cytosine and uracil bases in the primary transcript? So cytosine and uracil are pyrimidine bases. So in primary transcript of mRNA from this DNA, it must be remaining ones, 60%. So that's pretty straightforward. Okay. So coming back, which statement about xylem vessel is correct? Hollow enables the constant movement of water up and down. Constant movement up and down? No. Pits in xylem water moves in one direction, up, not down. So pits enable movement sideways, yes. Vessels contain numerous mitochondria? No, they do not. Vessels contain perforated cross-wall? No, they do. Ammonium ions in H4+ can enter xylem vessel by routes by two pathways. In apoplastic pathway, ammonium ions can move through cell walls until the pathway is blocked by the Casparian strip. Ammonium ions then enter the cytoplasm of the root cells. In symplastic pathway, ammonium ions move to the cytoplasm of the root cells. So there's nothing new here, I guess. A scientist measured the uptake of ammonium ion to xylem. This SGN mutation of A. thaliana, okay, does not have a Casparian strip. In this mutant, ammonium ions can enter xylem without entering the cytoplasm of the root because the Casparian strip is not there. The bar chart shows the scientist results. So uptake of ammonium ions, you can see in the non-mutant and mutant type. So there is this is the mutant one is not having the Casparian strip. So you can see there's a low concentration of ammonium ions. So uptake was still there. In the other plant variety, if the variance was compared, so again you can see that ammonium ion was absorbed, although this lacked the Casparian strip. Which conclusion can be drawn from this given data? Fewer ammonium ions enter the xylem when they have to move through the cytoplasm of root? No, more of them will enter. So more ammonium ions enter the xylem at low soil concentration of ammonium ions? No, more enter in the high concentration. Okay, the Casparian strip acts as a barrier to reduce movement of ammonium ions into the xylem. Again, this is a fact, yes, but there is no evidence on the basis of this graph. The loss of Casparian strip has little effect on the movement of ammonium ions into the xylem. Yes, this is exactly what's happening here. You can see that the non-mutant and the mutant, they have a very small change in how much amino ions are being absorbed. So the change is not very significant. So yes, D is correct. Here, we are not talking about the facts. Here, we are using the data to address the answer. Which row correctly shows the process required for the movement of water from a root hair cell to atmosphere? So cohesion, yes. Diffusion, yes. Evaporation, yes. All the three processes must occur. Which process is carried out by mature red blood cells? We know mature red blood cells have ATP in the cytoplasm. They can carry out the sodium potassium pump. They have that on the membrane, so active transport is possible. Yes, so active transport is the answer. Cell division, phagocytosis, protein synthesis are not all correct. None of them can be correct. Because it's like nucleus, so no cell division. Phagocytosis, again, it cannot engulf. There's no lysosome inside either. In protein synthesis, again, you do not have the a mature blood cell will not have the machinery required for protein synthesis, like no nucleus, no rough PR, no Golgi in the mature blood cell. The photomicrograph showed three white blood cells labeled X, Y, and Z. So just recall, Z is a monocyte, Y is a neutrophil, X is a lymphocyte. So which row correctly identified this? Simply B. Straightforward. When a blood vessel is damaged, it can result in protein problems in the blood body, which will identify the blood vessels that could have been damaged. Blood is not being distributed to organs that need it. So, would it be elastic artery? No. Organs are supplied with blood through the muscular artery, so either the answer is C or D. Elastic arteries are outright incorrect. Blood is not returned to the heart is collecting in organs. So, blood is returned to the heart through veins. So, yes, the answer could either be B or D. It could not be A or C. So with this, we omit out C as well. So the answer is D. Blood surges mean that relatively constant blood pressure is not maintained. So blood pressure maintenance because of blood surges coming in is the function of the elastic arteries. So yes, option is D. The diagram show how tissue flow is formed. This is the arterial end, this is the venule end, this is the capillary shown direction of blood flow. They are showing net capillary flow outflow, tissue fluid formation, inflow, tissue fluid reabsorption. These two arrows, one with the dashed line and one with the dotted line, represent blood pressure and the water potential respectively. Here you can see that this is the change in this blood pressure. So plus 1.1 kilopascal and plus 4.3, we calculate the difference here. This is the difference. This is the pressure already here. This is the pressure already here. So the difference is plus 3.2 kilopascal. So this difference will allow the blood to like move out into the tissue spaces. Similarly, this is the pressure change that can allow it to go back. So what I have done, I have just calculated the pressure changes which are there. These are the pressures existing within the capillaries, and these are the set of pressures existing outside the capillary. So if you just subtract each from its counterpart, we get the difference, which is responsible for either moving the fluid inward or outward. Now, what is the net pressure causing tissue fluid to flow out of the capillary at the arterial end? So out of the capillary, what would we do? We just would simply we just what do we do? We have this plus 0.32 kilopascals, and we have this this value of minus 2 kilopascal. What we'll do? We'll just subtract this from one another. I have pointed an static. I mistakenly pointed it here. So yeah, these are the two pressures that are responsible here. So plus 3.2 kilopascals is there, and I have 2 kilopascals provided to me here. I'll separate both the values. My answer comes out to be 1.2 kilopascals as positive. So the answer is C. The positive and the negative values which are shown here in this diagram, they only represent directions. All this stuff is not in our syllabus, but this is a question that came from a session objective 2. So you should be able to handle such questions just using some relevant logic, which I think is not very difficult in this case. What happens when carbon dioxide is transported by red blood cells? Carbon dioxide being transported by red blood cells. Hydrocarbon ion move into the red blood cells? No, they do not move in. They are produced inside. Chloride ion move into the red blood cells? No, they do not, and they do not bind to hemoglobin. Okay, so bind to hemoglobin is not correct. Chloride ion move into the red blood cells? Yes, they do. Hydrogen ion move into the plasma. Hydrogen ions do not move in plasma. HCO3 ions actually move into the plasma. H plus ion is left behind, which binds to the hemoglobin. Chloride ions move into the red blood cell. Yes, hydrocarbonate ions move into the plasma. Yes, that's correct. So D is the correct option. Simply a fact-based question. What is it typically found in trachea, bronchi, and bronchiole in the human gas exchange system? Cartilage is present in both, but not in bronchiole. Ciliate epithelium is present in all. Squamous is only in the alveoli and alveolar duct. Squamous endothelium, there is nothing like squamous endothelium. The term just doesn't exist. The photomicrograph shows a section through a part of the human gas exchange system. Okay, so what do we have here? We have this lining here. This is the columnar ciliated epithelium of the gaseous system, and this structure here, this is the cartilage, because the cartilage always have this appearance of having, you can say, gelatinous structures or consistency that shows some kind of secretions being accumulated around the cell. So this entire structure is the cartilage. This is how you identify the cartilage. And this is the muscle layer. These are the smooth muscles which are aligned, you can see in this direction. But anyhow, what structures are shown? Is it an alveoli? No, it cannot be an alveoli. Is it a bronchiole? It cannot be a bronchiole because the cartilage is shown there. It could either be a bronchus or trachea. Is it a bronchus or a trachea? It is not trachea, it is bronchus. Why? Because you can see that the cartilage is thin. The cartilage is not very thick here. Plus, these foldings in the ciliate epithelium are actually evident of this as well. Which factors are required for the efficient diffusion of oxygen and carbon dioxide in the human gaseous system? Clean and warms the air entering the lungs? No, there's no relevant function as far as the efficiency is concerned. Maximize the area for exchange? Yes, minimize distance between the alveoli and the blood. Yes, that's correct. So 2 and 3 are correct, whereas 1 is wrong. So again, using the information, if you are able to omit out 1 selectively, so you can be very sure that option A, B, C are incorrect. Only option D is the right answer. Which statement correctly identify why is it difficult to eliminate TB? Humans have limited access to clean water. No. No. Why no? Because TB is not a waterborne disease to begin with. Humans can be affected, but pathogen can be inactive. Yes, 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 because there are many people who are just hosts of TB, and in the sense that they are asymptomatic, they become the carriers and they transmit the pathogen to others, and in other people, the pathogen can become active. A number of drug-resistant strains have evolved. Yes, MDR-TB, XDR-TB, we have discussed this. So these two options are correct. So again, 2 and 3 are correct. Rest are wrong. Again, you can see the same thing. 1 is incorrect. So all the answers having 1 as an option cannot be the answer. Well, that's not always the case though. So you have to apply proper logic there. Actually they do this to confuse the students. So if like for example some students have a habit of okay, A1 is there, B1 is there, C1 is there, it means 1 must be correct. That's why they are given it in 3 statements. In fact, it's exactly the opposite. Bacteria can become resistant to antibiotic. What can help reduce development of antibiotic resistant bacteria? Using specific antibiotics to treat wide spectrum? Yes, the specificity is required. Using antibiotic to treat viral infection? No, it would promote the resistance because the antibiotic was not required. Developing new antibiotics? Yes, that's actually relevant as well. 1 and 3 are correct. Again, you can see 2 was given as an option in 3 of these, A, B, and D, and that's incorrect. Well, that's not always the logic. That's not always the case. So you have to go through every point very carefully. The diagrams show one way of testing the effect of antibiotic on bacteria. So this is colonies of bacteria. Petri dish with nutrient agar of bacteria. This is a filter paper antibiotic soaked. Is placed for 5 days. You can see the bacteria fail to grow around the region because of immunity. Because this is because of diffusion here. The antibiotic diffused in the surrounding. It killed the pathogen. It did not have resistance against it. So that's why it could not survive. The tables show the results of the test of testing 5 different types of bacteria on the same antibiotic. Zones of less than 13 millimeters show presence of the resistant bacteria. So if the diameter is greater than 13 clear diameter, it means that there's a resistant bacteria and they're all sensitive. But a diameter of less than 13 shows that there are resistant bacteria and they were able to grow in the presence of the antibiotic as well. Now, coming to the data. Types of bacteria are provided to you 5, 1, 2, 3, 4, 5. Type meter zone in millimeter, day 1, day 2, day 3, day 4, day 5. So these are 5 days on which they tested it. The first type of bacteria, this is the clear zone. You can see that the clear zone stayed greater than 13 millimeters, so they are all sensitive. In the second day, you can see a trend. Yes, but by the end, the diameter decreased, so less than 13. So resistance strains were there. Again, in bacteria third, this is you can say are being killed, but these are all values smaller than 13. It's similarly in 4, this is greater than 13, but this is greater than 13, greater than 13, but the last two values again are smaller than 13. In the fifth bacteria, all values are greater than 13. So this type of bacteria 1 and type of bacteria 5 are vulnerable to this antibiotic as far as this information is provided to us in this data. And the others, 2, 3, and 4, by the end of day 5, they are resistant. They do carry resistance. Which statement can be supported by by this data? All the types of bacteria become resistant to antibiotic over time? Maybe yes, but not supported by this data at least. Only type 2, 3, 4 bacteria show resistance to antibiotic. Yes, because they are the only ones whose diameter was less than 13 millimeter. The antibiotic can be used to treat type 1 and type 3 only. No, there's no such relevant information. Type 5 of bacteria can never become resistant to antibiotic. Now, how is that even possible? There's no such in data. Every bacteria can become resistant. It's just how they can exchange their plasmids and they become resistant. Some responses made by cells immune system to the pathogen are listed. Mitosis, recognizing pathogen, producing memory cells, secretes enzyme. Which responses are correct for phagocytes? What do phagocytes do? Do they undergo mitosis upon combating the pathogen? No, they do not carry out mitosis once combating the pathogen. They recognize the pathogen, yes. They produce memory cells? No, phagocytes do not have any memory cells. They secrete enzymes? Yes, they do. So 2 and 4 are correct. Some vaccines do not contain antigens. The vaccines contain a molecule of mRNA. Serve the immune system use mRNA molecule to make a protein antigen. The statement describes the stages of how mRNA vaccines work when they enter the cell of the immune system. So this is what's happening. B lymphocyte T lymphocyte, the complementary receptor bind to the protein. Okay, lymphocyte differentiate from memory cells, give long-lasting memory cells. This is the last thing to happen. Ribosome translate mRNA molecule to make a protein. Yes, because because you are producing a protein inside a cell. So the first thing the ribosome should translate. In fact, the first is the mRNA goes in. So then the ribosome translates it into the protein. The cell displays protein on its cell surface membrane. So what is the correct order? So this should be the first, this should be the last. It's a very straightforward question. So 3, 4, 1, 2 will be the correct answer. So that's how I hope this video was helpful for you. What I would recommend is that you try to attempt this question paper yourself. Now just pick up this one and start attempting it yourself and try to figure out if this video was helpful for you or not. So have a good day. Take care. Bye.